Good morning. It's my pleasure to talk about the Semiconductor and Health Science Initiative here in Taiwan. You cannot be in Taiwan and not talk about semiconductor. That is the island of semiconductor with excellence in many aspects of the health, semiconductor, and now one of the greatest trends in semiconductor, which is AI. And they are all combining together in a very unique time to make an impact on one of the most compelling topics of every society that we have around the world. And these are the numbers that speak for itself. We are dealing with a situation that just in the United States, about 20% of the entire GDP is spent on health science. That means in an economy of about $30 trillion, it's about 5 to 6 trillion spent on health and healthcare, and still there are a lot of opportunities to improve the quality of the health, and the same situation all over the world. So the first part of my talk, first half, I'm going to talk about how semiconductor progress and what has been accomplished in semiconductor industry can impact health in a very unique way moving forward to address some of these issues. So let me start with a couple of comments about semiconductor. As I said, in Taiwan, you have to talk about semiconductor, and that's where my background is. For many years, we really enjoyed the train of Moore's law, scaling transistors and offering more and more capabilities. And at some point, we came to conclusion that in addition to scaling, we have to have many other aspects of innovation, and these are examples of that. For example, when you talk about biosensors or analog or photonics, you need new materials beyond silicon, in addition to silicon. For example, in quantum computing or even in integration of the 2D materials with CMOS, we see many opportunities that can impact biosensing and medical and health science directly, and I'm going to talk about a couple of those in my talk today. Also, I want to highlight a very interesting, this quote from Heisenberg, who was the father of quantum physics, is very telling for me and exciting. He says that atoms and particles are not real, they are just possibilities. It's not a spiritual topic. It's about the fact that there is so much you can do with new materials. And just give you an idea how we have taken advantage of this thing. In 80s, we had a handful of materials in semiconductor. And now more than half of the periodic table is being used in semiconductor industry. And that was from 2020. Actually, right now, it's about even much more than just half of the periodic table. So I think the next innovation in semiconductor, or the most groundbreaking innovation in semiconductor, is new materials. I'm not saying that silicon is going to go away anytime soon for foreseeable future, but in addition to silicon, we see new materials can change the world in every aspect and many applications. Also, what I want to highlight is that it's not just the chip, but also the packaging, advanced packaging, and the architecture that can make a difference. Because in many applications, you have to bring multiple technologies together to accomplish things from AI processing to biosensing to RF transmissions and so on and so forth. So here's an exciting part. With all the advances that we have accomplished in semiconductor, now the sizes that we are dealing with semiconductor are very compatible with what we see in chemistry and biology. For example, some of the technologies that we are working on, how can we offer a readout of some biomarkers by putting the molecules on top of the transistor and then measure different properties or different circuit characteristics? How do we change the charge or mass or optical tag or magnetic tag or other properties or electron affinity and then measure it with very high performance circuits? So there are many examples of that. Actually, maybe Joel is going to talk about some of those, but we have already some accomplishments that is very exciting and very promising. Another example is what's happening in spectroscopy. Photoacoustics is one of those areas that is very promising and could open a lot of doors in the personalized healthcare field. And that requires taking advantage of silicon photonics, laser integration, and advanced MEMS technologies. When you get to the finer nodes or highest level of lithography, we see opportunities to even do protein sequencing using nanopores. And for that, you need to have EUV lithography. I think EUV is a very holy name, I guess, in Taiwan, because you are one of the leaders of using EUV for these advanced materials. And we think we can use that for medical applications and detecting molecules as well. And I'll show some example of that in my talk later. So semiconductor advances, one of the magics of the history of humankind, can make things much more efficient and much more effective, but not necessarily increasing the cost. The same magic can help us in combination with what we see in life science. Alex talked about some of that. Now we know how to do protein engineering a lot better than past, how to manipulate molecules. And with combination of these two, we are going to generate massive amount of data. And when you have so much data, AI comes to the rescue. So combination of these three and confluence of these three areas opens a new opportunity for us to handle personalized healthcare in a different way. So semiconductor technology has done a lot for us. Look at the phones in your pocket or in front of you. It used to be millions of dollars and a power of supercomputer to offer the same processing power in your phone, and now it's a few $100s. So a supercomputer ended up being in your pocket, supercomputer of 30 years ago or 40 years ago. I remember I designed this radar system as part of my PhD for synthetic aperture radar chip for a defense company, and it cost millions of dollars. Now TI has a chip for $10 doing the same thing. So you can imagine we got almost 1 million times better in terms of cost efficiency in semiconductor. We can do the same thing in medicine. So you can see that now you can read the glucose on your body at clinical level, thanks to some of the advances in semiconductor and biochemistry, or pills that can do this endoscopy in your body, disposable pills, and insulin pumps and neuropixels. These are magics that are happening, and it's just the beginning. Here are some examples that some of which we are working on, some of which I've worked on in the past, and we see the opportunities that bring molecules and atoms on semiconductor and offer opportunities for diagnostic and intervention. For example, using microneedles for drug delivery or targeted drug delivery, or smart fabrics to measure the performance of the muscles and joints and react to therapeutically to heal the muscle or joint. These are technologies that 10 years ago was just a dream, and now we see a huge opportunity for us to offer these technologies at the scale. So the vision that we have for the future of the medicine is that, or personalized medicine, is that today, very small percentage of diagnostic and therapeutic protocols are based on understanding of the molecular performance of the body. Most of them are based on, here's the symptom and here is the therapeutic solution for that. And I heard that most of it, I mean that 5% or 10% is mainly in oncology. So what we wanted to change about that is by offering a clinical quality continuous monitoring of the molecular biomarkers and physical biomarkers, change that picture by offering massive amount of data, quality data, about how your body works, and use AI to translate these to a more scientific approach of disease. In other words, the machines in the lab, which today analyzes your blood and biomarkers in your blood and plasma, we wanted to bring it to the point that you can continuously monitor these with the same quality. And again, you say, is it possible at all? We have done it once, and it's been very successful. Continuous glucose monitoring today, which has about 40 million users in US, and I was heavily involved in developing that about 12 years ago, showed that you can get exactly the same quality of the glucose in the blood as you get it in the lab, but the difference is on a continuous basis. So you can see the trends, you can see the correlations, you can see what other biomarkers can impact each other, and translate it to opportunities for a better diagnostic and therapeutic approach. Also, we think that in the near future, we will be able to do single molecule counting and characterization at the point of care. Hopefully, a device the size of your phone can offer the protein sequencing that today we are getting with a mass spectroscopy machine in the lab. And these are opportunities that can change the whole picture. Continuous monitoring with much smaller sample, continuous monitoring before it gets to the point of symptomatic, and also profiling proteins in the population of single cell, not averaging over a massive number of cells, and also closed-loop drug discovery and many other opportunities can be enabled by these kind of technologies. Of course, it requires a very close collaboration between clinicians, semiconductor guys, and statisticians and data analytic guys to translate it to some actionable diagnostic and therapeutic profiles. So just a summary of technologies that we are working today. Electrochemical sensing is very promising. These are the extension of what we are doing today for glucose profiling. We can do it today. Actually, I was showing Alex the other day, we can do it for lactate, but also we can potentially do it with cytokines and with many other small or low concentration molecules. We are working on advanced ultrasonics. Ultrasonic is the magic of the future. I think there is a lot we can do with ultrasonic because it's very non-invasive, at the same time could be high precision. In fact, there was a paper from Nature, I just saw it a couple of days ago. You can get the resolution almost at the level of MRI, and in some cases better. And it's going to be a functional MRI, meaning that you can get the same performance during the daily activities. Same thing for magnetics. Can you imagine if you can monitor your brain waves while you have your daily activities? That can change the whole scope, because today, to measure brain waves, you have to be in a highly shielded room, because brain waves are about a few femtotesla per root hertz, whereas Earth magnetic field is about 100 to million times stronger. So that's why you need to be shielded from everything else. We think in the future we can have the kind of technology to monitor brain activities during daily activities and translate it to some diagnostic and opportunities for detecting disease. The first one is epilepsy. We are working with Harvard team on that, but we see that dementia and other neurodegenerative diseases are also candidates for that. And also photoacoustics. So the future we can imagine is clinical quality molecular biomarker and physical biomarker continuously measured. Massive amount of data gets translated to some therapeutic protocol. And therapeutic protocol is not just drug delivery, of course includes drug delivery, but neuromodulations, smart fabrics, and many others. I mean, I can go through the list of some of the neuromodulation techniques that we are looking at, and it's extremely promising. In fact, some of them are FDA approved already. If you think it's too far out, we have already a closed loop today. Today, based on CGM measurements and analyzing data, we can control the amount of insulin, how much and when, and it varies for one person to the other. Why can't I do the same thing for many other diseases? Of course, glucose is a big molecule, and it's been around for a while in terms of research, so it was the first good target. And guess what happens? You start from clinical quality, you end up with wellness. Now people have these patches for quality of life to make sure that their diet and sleep and activities are properly adjusted. So we see opportunities both at clinical level, but also at the wellness level. So AI is a big part of this story. It's almost influencing and impacting many aspects of the healthcare. I'm not going to talk about most of them. I'm not a biologist, so I'm going to stay away from heavy-duty biology. But I would say that on robotic surgery and some of these genomics and then potential drug targets, we are working closely with biologists and clinicians to use the semiconductor capabilities and offer solutions for these technologies, for these approaches. So with that introduction, the second half of my talk, I'm going to talk about two technologies that are very promising, very exciting, and we are making good progress on that. One, again, all of this that I'm talking about is at wearable or point of care form factor, yet clinical quality. So it's not a smartwatch. It's something that you can read the measurements and translate it to a treatment plan. So first one is what we call it photoacoustics. Photoacoustics is about how to do a deep tissue spectroscopy, deep tissue, with high chemical selectivity and equally important spatial resolution. Photonics by itself cannot do it. For those of you who have worked on photonics, you know that when you shine a laser to the body, reflection is getting scattered by the skin by a lot of factors, and by the time that you get the signal, it's so low in terms of signal to noise, literally cannot penetrate meaningfully in the body more than a few microns. So we thought that, okay, how do we fix this problem, improving signal to noise, yet use the benefits of the laser, because selectivity and the bandwidth that you can cover in laser is very important for a lot of biological aspects of it, because you can tune the wavelengths to the resonance frequency of the molecule, especially CH bonds, and then detect molecules that have high interest. So okay, why don't we combine laser photonics with acoustics? And I'll tell you in a second why. So the idea is that how to convert the molecular vibration, I'm talking about vibrations at resonance frequency, not rotational or electronics, into acoustic waves to detect molecules and chemical components in the tissue. So the concept is the following. First, you make sure that the incident photons frequency matches the resonance frequency of the transition frequency of the molecule that you want to detect. Now, the first reaction for engineers is that, okay, you send photons, you detect photons. No, we send photons and we generate heat because that specific molecule, hopefully, absorbs maximum energy because the frequency match with resonance frequency, and there is a thermoelasticity, so it starts expanding. Now, as soon as it expands, we turn off the light, meaning that it's pulse modulation. And there, you have this expansion and shrinking, and that is ultrasonic wave. So you send photons, you detect ultrasound. Why is that? Because with sending photons, we can have selectivity. By receiving acoustic waves, you improve signal to noise. Now, what frequencies are good for CH bonds? We need near infrared, 1100 to 1300 nanometers. For hemoglobin, that's our first target. We wanted to take hemoglobin for GI tract and bleeding in the stomach, and we are working with Mass General Hospital on what kind of hemoglobin selectivity and sensitivity we need. That is where you need 400 nanometer and about 1.1, I'm sorry, it's micrometer, not nanometer. And the reason that we picked those frequencies or the wavelengths is because in those windows, the absorption of hemoglobin is higher than water because we have a lot of water molecules in our body as well. So we need to make sure that we get the right wavelength. So here's the concept in graphics. We modulate light. We match it with the vibrational resonance frequency of the target molecule. And here is the challenge. You need a super sensitive microphone to detect that waves. You cannot just take the microphone that is attached to my tie to use that, because you need at least 10 to 15 dB higher SNR, and I'll tell you how we achieve that. And then you have potentially a selectivity and sensitivity for clinical quality detection. So why ultrasound? Acoustic scattering is 3 orders of magnitude less than optical scattering. So acoustic signals can be very, very spatially accurate inside the body. That's why I love ultrasound. I mean, especially with MEMS ultrasound technologies that we are working on, we can do magic with ultrasound. We don't have time to talk about it. But in general, ultrasound is much more friendly to biological tissues. One thing that ultrasound doesn't like is air bubbles, because then the impedance mismatch is going to destroy the SNR. So we fixed that problem with a kind of elastomer that we can talk about in our social hours or social break. So in this case, unlike a typical smartwatch and other devices, it's a one-way photon travel. So photons travel one way. We don't care when they come back. We care about the acoustic wave. And when it's traveling one way, it doesn't have to be ballistic. You can gather entire energy as long as resonance frequency is matching the resonance vibrational resonance frequency of the molecule, and you can then detect the signature of the molecule. So as a typical science and engineering problem, it's easier said than done, because when you get to these molecules, it's not just one molecule in plasma waiting to be detected. You have a whole bunch of them, and their absorption frequencies are kind of like adjacent. So you need to do a lot of training to find those signatures across a wide band. You cannot just take 400 nanometer or 1.1 micron. I mean, ideally, that would have been fun, right? I mean, taking one frequency. So what we are doing is that we generate thousands of spectrum lines, detect these signatures across the board, and then map it to the molecule. But how do we do that without using a big Raman spectroscopy? So we used an idea of dual comb laser. The idea of dual comb laser is that you get a quantum cascade laser with indium phosphide on silicon, integrated it, and generate a wide range of bands, and then shift it and mix it together so you can translate it to ultrasound. So it's a really interesting way of mapping terahertz to megahertz without a mixer. So we use the second laser as mixer here. And we see it's very promising, and we think that so far the results, I mean, it's early to say or early to celebrate, but you can see that in this case, the integration of indium phosphide laser with CMOS signal processing is a back end of the line process. So we are not interfering with CMOS process. As I said, microphone is another challenge to deal with. So typical microphone, how does the typical microphone work? Either piezo or capacitive sensors. That is, detecting vibration to electric signal. That's not good enough. SNR is not good enough. So we came up with an idea of how to use photonics to detect sound waves. So here is just designing a new microphone. The idea is that if you squeeze the waveguide, you modulate the photons, and by detecting modulated photons, you can understand what's going on the acoustic wave, which is squeezing the waveguide. So you can see in this picture, the waveguide is hitting this optical waveguide, which is on a SOI substrate. I mean, you have to have an SOI, as you know, every photonics is on a box. And then this modulation is detected as a signature of the acoustic wave. So again, magic of semiconductor is putting a lab in your pocket. So it's not just a phone or computer in your pocket, it's the whole lab. It's either in your pocket or on your skin, depending on what you want to detect. And there are other approaches. We are looking at also aluminum scandium nitride as an opportunity for high quality piezo, and we are comparing these two performance to see which one is more interesting. As I said, these absorption frequency, resonance frequencies varies quite a bit. Water, hemoglobin, oxygenated, deoxygenated. So we need to go across different spectrums and do AI to translate it to enough selectivity and sensitivity for medical applications. So I have 6 or 7 minutes to talk about another interesting topic, which is very dear and near to people who like lithography technology, because that requires 2 to 10 nanometer lithography of the silicon to detect protein, one molecule at a time. So what we call it solid state proteomics. As I said, I'm not a biologist, but when I read about proteins, I was puzzled by the mysterious kind of world of proteins. I mean, a typical human protein has about 300 to 500 amino acids. So the combinations would be 10 to the power of 390. The entire atoms in the observable universe is 10 to the 80. So you can imagine how many ways these constituents come together and create different proteins, and each of them singing and dancing in a different way. So we are dealing with a very, very complex set of molecules. And interestingly, a single protein had tens or hundreds of modifications. So you can imagine 10 to the 390, and then each of them has so many different sites and modified sites. And I think having this capability of sequencing proteins today is based on, first of all, you cannot do single molecule. Second, you need these big machines. But then the question is, why do you need to have protein sequencing at the point of care? Why don't you send it to the lab and sequence it? Many reasons for that. First of all, you can do drug discovery a lot more efficiently. Pretty much you can see how body metabolizes these drugs and measure it rather than just looking at secondary symptoms. You can do drug discovery, you can do many other diagnostic and therapeutic techniques, closed-loop techniques, by having a real-time proteomics or protein sequencing and protein. Even in agriculture and food science, there is a lot of applications that you need to measure or characterize or sequence these molecules at the point of care or in a small form factor. So optical and mass spectroscopy is the common way. These are relatively slow. So we decided to use the magic of semiconductor to characterize proteins. And the idea is that we etch these tiny holes, the size is about 5 nanometer to 15 nanometer, in a piece of silicon nitride, and then let these proteins go through these holes. So you can think of it, a membrane with charges on two sides and a voltage applied. Of course, electrons go through these holes or these tiny pores under normal conditions. When these proteins clog the pores, they change the current profile. And that is where you can characterize protein. So here's the concept. So you can see the membrane. Yeah, my pointer gets lost in this bright screen. But you can see there's a membrane, and there is a voltage applied to sides of the membrane. And as the protein molecules get stuck or pass through translocation of these protein molecules through the holes, you see the characteristic of the current that you're measuring in the circuit varies. And by measuring these characteristics over many molecules, you can come up with idea what kind of constituents you have in protein molecules. So here is a more detailed concept. As these single molecule goes through, because the size of the pore is almost compatible to an unfolded protein. We are working on a folded protein as well now, but today they unfold it and pass it through the pores. The idea is that can we do more efficient way, and that is yet to be defined and understood. I'm trying to understand what it takes on the protein side. So that is the part that some of the government research agencies are very interested to characterize folded protein. So anyways, by passing the molecule through this pore, we can monitor the current profile. But it's an extremely noisy environment. So you can imagine that one molecule is passing through this pore, and it changes the transition of the electrons between two sides of the membrane. And that's why you need an extremely high precision circuit with extremely low noise figure to detect that kind of current variation, and then use some signal processing and AI to characterize it. So for those of you who are in semiconductor industry, I don't want to spend too much time. Here is the chain of process to create a nanopores. Again, if you are using EUV on 300 nanometer, 300 millimeter wafer, and the pore size is about 3 nanometers all the way to 15 nanometers, depending on what kind of sensitivity and molecules you want to detect. It's a 10-step process, and this already has been tested. We have some results, and here are some results before showing this thing. So here's the setup. There is already biopores already, and biopores are interesting. The problem with biopores is not as consistent as solid state, and that's why we think solid state pores would be more promising. And we need to have data statistics or machine learning to analyze this data and map it to the level of selectivity and sensitivity we need. The picture you see here is the real work that we did with IMEC on designing these nanopores. It's the size of smaller than your laptop. So you can have protein sequencing on your desk. And I think next step would be protein sequencing potentially in your pocket. And by the way, you're going to have tons of these pores on the silicon, but amazingly, these pores are, let's see if I can move my slide here. Yeah, Skip that one. So here are some results we have, some measures. So you can see that when you put it to different RNA ring, DNA cube, and fragments, we can detect it based on the characteristic of the current that you measure across the membrane. Same thing with 2 types of proteins. We have tested this, and these are all promising and showing that we can do it. And also, we are working on jointly with a Swiss company, MIT, and IMEC, on using nanopores to make these pores addressable. So you decide which pore gets activated at what point. And that also shows more control over these advanced proteomic machines. So with that, I'd like to thank you for your attention and wrap up my talk.